everyone, Mrs. KJ here. I wanted to go over the student guide looking at cells through a microscope because there's a lot of instructions and I didn't want you to feel overwhelmed. So first of all, you want your materials that are provided, the cover slips, which are the little tiny glass pieces, the microscope slides, and your microscope. The one item that is required that wasn't provided is the light for the microscope. So I'm holding my microscope here on the arm. What works best is if you have a cell phone with a light on it and you put it right underneath. See how it's illuminating the prism there? And you want to make sure that when you're looking through the eyepiece that you can see a nice light circle. So try that first, because if it's not a nice light bright circle, it's not going to work. It's not going to be enough light to see through the layers of the plants and whatever else you're testing to see the cells. Using a cell phone, I suggest making sure it's on the highest light. So if I hold my cell phone, if I hold the button for the flashlight, I can put it all the way up and that makes it the brightest. If you have a different LED light or a flashlight, that will also work. For what to try out, it's whatever you have around the house. We have some ideas here for you, but don't go out and buy anything. The first thing listed is iodine. That will stain your cells so you can see them more easily. However, I did not have iodine and I could see everything on this list, so it's completely up to you. I'm not going to read through absolutely everything, but I want to do the highlights. So your cheek cells. This is what your cheek cells are going to look like, except they won't look blue because you're not blue. This one has a blue stain on it, but I like this image because it really showed you that the cell membrane kind of looks like a halfway deflated balloon. And you can definitely see the nucleus in a couple of your cells. You're not going to see them in all of them, but a couple of them you can. To get your cheek cell sample, take a toothpick. Do not use the pointy end. You want to go flat like this on the inside of your cheek, like this, so that you can see that I'm pushing out, but it's not painful. I'm just rubbing it. And then you're going to see like just a little drop and maybe a little whitish. That's what you're going to put on your slide and then put a cover slip over top of it. Very important with microscopes, you need to get it in focus at the 4x, get what you want in the center, and then switch to 10x, get it in focus, then switch to the 40x. With 40x, you should barely be moving your focal points, because if you move it too much, you'll actually break your cover slips. So be careful with those. One other thing I wanted to mention about the cheek cells is you'll have a little bit of saliva on it, and when you put the cover slip on, when you're looking through, you might see the saliva pushing, and it looks like it's pushing little chunks on 4x. Those weird shaped little chunks are your cheek cells. Your cheek cells are not perfect spheres or cubes. They kind of look like little blobs. They're called squamous cells. Next one is the onion. There's two different parts that you want to check. The first part I have in my picture here is the very outer layer that feels papery, and you want to get it as thin as you can. With that, I could definitely see some cell walls, but because there were so many layers of cells, it was hard to really see much more than that. I then took a piece of the inside of the onion and I very carefully peeled it. So you can see here it almost looks like plastic. That's one of the layers of the onions. So you want to definitely look at this plasticky looking part. It's obviously not plastic, it's part of the onion. And then the inner onion too. So you can definitely see some cell walls and possibly vacuoles or nuclei. Yes, onion's a plant, but you also want to try a green plant. I personally like succulents because when you peel an edge off, it gives you, again, that thin layer that feels like a thin plastic. And you can check that out, and you can check out the inside. Succulents are really neat because you can really see a vacuole here or there. Again, you're not going to see them in every single cell, and you're not going to see the nucleus in every single cell because there's many layers of it. And the microscope can only focus on one layer at a time. However, with 40x, I definitely saw some chloroplasts, and even on 10x. The little green dots are going to be the chloroplast. For some bacteria, what I find is the most exciting is if you can get some pond water. So you want to get some algae or some floating little bits in it. Sometimes if you're lucky, you will see some organisms. You might see an amoeba that looks like a skin cell and can actually be going around something else to eat it. You might see something moving around with little hairs called cilia, and you might just see some algae. But definitely if you have something nearby, check it out. If you are thinking to yourself, I live in the city, there aren't any ponds or lakes nearby, there's probably a good drainage ditch or something like that. So make sure you ask your parents before you go. But there's probably areas where algae is growing in water. So check it out. Hair. Hair is very interesting. If you have long hair, what's neat is you can actually look through while your own hair is on the stage. But get some hair, put it down. If you can get a long enough piece that you actually put it through the clips, I found that the easiest, so it stayed where I wanted it. You're not going to see the cells, but you're definitely going to see the edges and some contouring of the hair. It's really interesting if you can compare it to somebody who still has pigment in their hair versus if you have a gray hair. The gray hair is going to look different. Curly versus straight hair, or if you're able to use a hair from someone of a different ethnic group. That's cool to check out too, as well as just different colors. Even if everyone in your family has exactly the same hair, still check it out. It's kind of interesting. Other things, it was brought up, you can try yogurt or probiotics, or you can use your toothpick and scrape some of the bacteria and scum out of your teeth. But those things are so small, you're not going to see more than little dots. And the same thing for blood. Someone always asks about blood. Your blood cells are so much tinier than your cheek cells. Yes, I totally did try it just to make sure. All you see are little dots. It's not worth it, in my opinion. Cleaning your slides. So you don't want to scrub them because you can break them. Just get like a small dish about this big, put it with some dish soap, hot water, put your slides in, let them soak, wash them off, and then just pat them dry and let them air dry before you put them away. Questions to think about before you take the quiz. So let's say you didn't get through all of these, that's okay. Let's say you only got one to work, that's okay. And if you want some help with your teacher, definitely ask so we can help you because it is difficult. It's a skill to use a microscope. But these are the kind of questions that everyone should be able to think about and answer even if you didn't get all of them to work. So which of these cells on here were animal cells? Which one were plant cells? Which ones were single-celled organisms? What structures could you see? So for example, if I mentioned chloroplast, where would you see that? If I mentioned a nuclei, where did we talk about that? Or a cell wall or a vacuole? Those are the big ones that we talked about. And what differences did you see between plant and animal cells? If you have any questions, ask your teacher. Have fun. Bye.